When we're looking at the fossil record through the microscope, we only find the most robust species. There's lots of organisms that do not have a robust skeleton and they don't fossilize. And so the aim of ancient DNA techniques really is to look at the broader picture of those species as well. My name is Armbrecht and I work as a postdoc at the Australian Centre for Ancient DNA at the University of Adelaide in Australia. The field of ancient DNA is primarily been used to study human evolution or megafauna evolution, and the application to marine environments is relatively new. We want to avoid any type of contamination because ancient DNA is very, very fragmented and degraded. In contrast to that, modern DNA is very abundant and intact. So if we're introducing any bit of modern DNA into our sample, this can override our ancient DNA signal. So there's certain things that we can do to avoid contamination during sampling on a drill ship. We're using piston coring, and then what we can additionally do is to introduce a chemical tracer into the drill fluid. And then the core comes on deck, and this is where we can really make a difference. We have a lab coat and gloves, goggles, and a face mask, and we're establishing this barrier between us and the sample. If I wasn't taking all of those precautions, the danger is that I might find the DNA of myself or DNA of anybody who's helping me to handle the cores, rather than the DNA that I'm actually looking for. I find phytoplankton fascinating because they're very important organisms. They produce about half the oxygen that we breathe. They're very good climate indicators. They're also the base of the marine food web. We can look at how communities change from warm periods to cold periods, and that can tell us about the past ecosystem and how the marine environment looked at that point in time. By using ancient DNA, I can look into the past marine ecosystem in a more complete, comprehensive way.